Welcome back, ladies and gentlemen. So today we're going to continue to work on the meiosis drawing worksheet, and we're going to pick up today with meiosis 2. So that being said, let's take a second and go backwards and take a look at what was going on at the end of meiosis 1. So end of meiosis 1, although we didn't draw these diagrams, when we talk about telophase 1 and cytokinesis 1 to end that first round of meiotic division, we're talking about taking that diploid cell, splitting the cytoplasm during cytokinesis 1 into two daughter cells, both of which are now haploid because those homologous chromosomes, this guy here and this guy over here, are now going to be in separate cells once this division of this cleavage furrow finishes. So those two resulting cells at the end of meiosis 1 are both haploid, and neither are identical, not only because those homologous chromosomes are no longer in the same cell, but also because the information on those chromosomes has also been mixed up because of crossing over, as well as that independent assortment that happened during meiosis 1. Now, when we look at prophase 2, again, there's no specific place here to draw this. That being said, you can see the diagram there at the top, and this would also be similar to what you would see during interphase 2, which really is minimal if it ever really exists at all. There's some debate on that, because once we roll from cytokinesis into that second round of division, we're not going to go through any of those G1 or the S phase or the G2 again, because these cells are already having the information duplicated in those sister chromatids, right? So these guys are already here. The only point to meiosis 2 is to separate those sister chromatids and finish that division through the second round of division. So there really isn't much interphase at all, but if you were to have it to sort of just arrest or stop the cell in that cell division there, it would look very much like this here. So we're not going to bother drawing either of those. That being said, if you want to, go for it. But under prophase 2 that is labeled on your worksheet, we are going to put a couple of notes there. And so remember that by this point, we're already haploid. Those homologous chromosomes are in separate cells. Okay, and during prophase 2, it's going to look a lot like mitosis. The nuclear membrane is going to disappear, the nucleolus is going to disappear. Okay, your centrioles are going to reappear on either end of the cells and start migrating to the opposite ends or the opposite poles. Spindle fibers are going to be attaching from those centrioles to the centromeres on your chromosomes. And this is happening in two cells at one time. So this is happening simultaneously in both of those cells that were created at the end of meiosis 1. Okay, we are going to draw metaphase 2. Okay, and again, if we take a look, this is still haploid. You're going to have spindle fibers that are going to finish attaching from those centrioles to the centromeres of the chromosomes, and those chromosomes are going to line up at the middle of the cell, but now we've got two cells happening at the same time. All right, and so if I draw this out, okay, so here's one cell, right, and we've got partially that crossed over chromosome here. Okay, so we're going to keep tracing that out. And then we've also got on this side its homologous pair that also had the crossover. Okay, so you've got those guys. And then we didn't show the crossing over in the other sets of chromosomes, although it could have happened, but we're not going to show that, so we're just going to draw those guys in here. Okay, now we're still going to keep track of certain structures and labeling those structures. So if we take a look, all right, we still have those homologous chromosomes, but note how they're no longer in the same cell. Right? And that's really important because that's what now makes these two cells haploid, because those homologous chromosomes aren't together anymore. Now your sister chromatids are still attached, so we'll label one of those. All right, and they're still attached at that attachment point in the center there, that's your centromere. You've got your centrioles out here on either ends. All right, and from those centrioles are where your spindle fibers are originating from, and they're ultimately attaching at that centromere. Okay. And if we take a look, right, these guys are lining up in the middle of the cell. And so we've got this sort of imaginary line that goes between each of these cells now. All right. And that imaginary line is what we call your metaphase plate. Okay. So each of these haploid cells has all those spindle fibers attaching from those centrioles to the centromeres. Chromosomes are lining up along that metaphase plate in the middle of the cell. And again, we're still going to have another round of checkpoints here to ensure that all of those spindle fibers are attaching to those centromeres. So then we get to anaphase in the next round of division, that those centromeres come apart like they're supposed to, and that each of those sister chromatids moves to an opposite side of the pole or opposite side of the cell so that that genetic information gets split evenly. If we blow through that checkpoint, here's another place where we can have a non-disjunction event. And when that happens, that non-disjoining, that staying together, those sister chromatids ultimately results in a cell that now has too many chromosomes and another cell that's going to end up missing them. And that's where we can have something like the event with somebody having Down syndrome. There was a non-disjunction event where we blew through this checkpoint in metaphase either 1 or metaphase 2 here, and those sister chromatids remained attached. That's how they end up with 47 chromosomes instead of the 46 that a normal functional cell is supposed to have. Okay, anaphase 2. Going to look again a lot like mitosis, so to save us some space and some time, we're not going to draw this out. But if you take a look at the diagram up there at the top, you can see how those chromatids are now splitting. They're moving along those spindle fibers to the centrioles at each end of the pole of the cells. All right, and so we're just going to add some notes there. Okay, this is a haploid stage, just like everything so far has been during meiosis 2. And then we can add the notes there that the sister chromatids are now what's coming apart. Unlike in meiosis 1, where it was the homologous chromosomes or the tetrad splitting, this time we've got those sister chromatids that are separating. Again, looks a lot like mitosis, but we're happening in 2 cells at once. All right, and that brings us to telophase 2. Again, still a haploid stage, all right, but your centrioles and your spindle fibers at this point have disappeared. Your nuclei are back, so the membrane's back, the nucleolus is back. It's surrounding those chromosomes. Those chromosomes that have been this tightly wound structure are now going to start relaxing back into chromatin so that your cells have access to them once we get past this stage. And this stage will overlap cytokinesis 2. So what you're actually looking at here in the diagrams is the end of cytokinesis 2, where we ultimately result in 4 genetically different daughter cells by the end of this round. So let's go ahead and draw in some of the structures and what this is going to look like. So I'm going to add in a nucleus. That's going to happen in all 4 of these cells. All of these cells have their cell membrane still intact. And it has been pinched off now that your cytokinesis has occurred. All right. But what's really important here is to take a look at what ends up happening with each of the chromosomes. So I end up with a full red one on this guy, but the other sister chromatid, remember, was half red and half blue. Right. Now over here we had the opposite. The top half was blue and the bottom half was red. And then that final cell was all blue. And then I still had those yellow and green ones as well, but we didn't do any crossing over. So this one gets all yellow, so does this one. And then those 2 guys at the end are going to get those 2 green ones. Okay. And so if you look at the coloration of all of these, each one of them has a slightly different color combination. This is red and yellow. This is red with a little blue and yellow. This one's blue with a little red and green. And this one's blue and green. So we can look at the fact that none of these cells are genetically identical. We've got this crossed over chromosome here and here, which causes us some difference. Plus, because of the independent assortment, we've got these guys going on in here with that yellow and that green. And so at the end of this, 4 cells, all genetically identical, or all genetically different, I should say. They're all haploid, so chromosomes are separated. And if you take a look at your worksheet, if you follow down the one set of lines, all right, the dotted lines are going to get you to the eggs. So if this were a female's body, then these would be the 4 different cells her body would create. And if you follow down the solid lines on your worksheet, that'll take you to sperm. And if this were male, then these would be the 4 genetic combinations that would be made in those sperm cells. So you can take those colors in terms of the chromosomes that are labeled here and follow those color combinations down to those eggs and those sperm and add them to that as well. All right.